Hello, everyone, and welcome to today's live broadcast, Understanding Cancer, Drug Delivery Nanoparticles Illuminated, the Light Scattering Toolkit by Dr. Daniel Sohm, Principal Scientist and Director of Marketing at Wyatt Technology Corporation. We're excited to bring you this educational web seminar presented by LabRoots and sponsored by Wyatt Technology. More than 40 years ago, Wyatt Technology scientists invented the very first commercial light scattering instruments, incorporating lasers as their light source. They have been defining and redefining the state-of-the-art laser light scattering hardware, software, training, and services to meet customer needs for over 3 decades. Along the way, they have added several related technologies to Wyatt Technology's product line, including dynamic light scattering, refractometry, viscometry, and fuel flow fractionation. To learn more about Wyatt, please visit wyatt.com. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. This presentation has been approved for continuing educational credits. If you want to obtain the credits, please click on the Get Your Free CME CE Credits button located in the lower left of your screen. It will take you through the process to obtain your certification. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. Finally, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. Thank you. Now I'd like to introduce Daniel Sohm, Principal Scientist, as well as the Director of Marketing at Wyatt Technology Corporation. He's been with Wyatt for over 11 years, first in R&D and then in the marketing department. Prior to joining the scientific instrumentation world, his professional endeavors included the semiconductor and defense industries. Dr. Sohm completed his undergraduate degree in physics at the Technion Israel Institute of Technology, his doctoral research in the Brown University Physics Department, and postdoctoral research at Los Alamos National Lab and the Weizmann Institute of Science. His publication list includes, among other topics, a book chapter, review article, editorials, application notes, and patents on the characterization of biomolecular interactions by light scattering. Please join me in welcoming Dan Sohm. I will now turn the presentation over to him.
Thank you, Judy, and welcome everyone. Glad to have you aboard. So you can see the title of my talk up on the screen. This summer, actually, I attended the Control Release Society meeting in Edinburgh, Scotland, and it was absolutely a terrific meeting, I'd like to say. I really enjoyed it, and I saw lots of opportunities and need out there for the kind of technologies that I'll be discussing in terms of characterizing nanoparticles for drug delivery. And so it's a real pleasure to be able to present the webinar today and perhaps discuss some of those opportunities with you. So the tools that I'll be discussing that are based on light scattering are used to answer a series of analytical questions. And first of all, the questions that you'll be asking are, what are the solution properties of the constituent macromolecules that come together to form your nanoparticles? That may be polymers or proteins, peptides that are perhaps encapsulated or adhering to the surface, as well as oligonucleotides if you're doing siRNA delivered by liposome. And the properties of those macromolecules that we'll be characterizing with light scattering include molecular weight, oligomeric state, size, conformation, conjugation, for instance, if it's a copolymer or a protein bound to a carbohydrate, and charge. The acronyms that you see over here are some of the tools, the light scattering-based tools that we'll be discussing later on, and they typically include some form of light scattering. For instance, this MALS part is a type of light scattering, and DLS type of light scattering, and a sample preparation, for instance, SEC, which is used upstream of the light scattering to prepare and deliver the sample for optimal measurements. Another set of questions you'll be asking are the solution properties of the nanoparticles themselves. So what is their size, aggregation state, the conformation of the particles, binding of functionalized ligands to the particles, and their zeta potential, which impacts stability. And then finally, once you have a product, is it well behaved? So again, how does it behave over time? Does it aggregate? Is it stable? Does the drug which is encapsulated in the particle remain bound, or does it diffuse out into solution? And perhaps the reaction kinetics once it's combined with some reagent which simulates the physiological effects. Solution to this characterization can be found in light, actually, and so light will help us discover these properties, quantify them, and monitor them for optimal preparation. So the first type of light scattering or the first flavor of light scattering that I'd like to describe is called multi-angle static light scattering. And what we're going to do in MALS, or multi-angle static light scattering, is illuminate a solution with a laser beam, and there will be macromolecules or particles in the solution. They'll scatter light in all directions, and some of that light will reach a detector. And so we'll have a detector placed at a particular angle, theta, relative to the laser beam. And it turns out that this is actually a first principles relationship, that the intensity of the scattered light is proportional to the product of the concentration of the molecule, its molecular weight, and there's also an angular dependence, which depends on the size of the molecule, and that's captured in this P of theta term. And P of theta actually goes to 1 as theta goes to 0. So if we could place a detector right over here at 0 angle, this term would go to 1, but in order to analyze the size and shape of the particle, we need to place actually detectors at a series of angles in order to map out P of theta. The properties that can be determined with MALS include the molecular weight, or the molar mass of the molecules, and also a measure of size called the root mean square radius, also known as RG. Since this is a first principles technique, it can be used to derive some important parameters, for instance, the stoichiometry of association, binding affinity, rates of association, dissociation, and also a measure of interactions known as the osmotic zero coefficient. And so because it's first principles, we can in fact derive in-depth characterization using multi-angle light scattering. The second flavor of light scattering is called dynamic light scattering, also known as QELS. And similar experimental configuration to the one I showed you in the previous slide, except that now instead of measuring the average intensity as a function of angle, we're going to measure the noise. So if you examine closely the light that comes to the detector, you can see that it has rapid fluctuations. And those fluctuations actually contain important information. They're related to the Brownian motion of the particles. And so as the light beams scatter from the particles in solution, you'll have coherent interference of the beams as they approach the detector. And depending on the distance between the particles and the distance to the detector and the angle, you'll have scattering in and out of phase, which will cause these fluctuations in intensity. The rate of fluctuation is related to the rate of the jittering around in solution, the Brownian motion. And so therefore we can analyze that to determine the diffusion coefficient. And once we know the diffusion coefficient, we can add in a few more parameters, temperature, viscosity, and so determine a different measure of size called the hydrodynamic radius RH. So dynamic light scattering, or DLS, from first principles gives you the diffusion coefficient, from which we can calculate an effective hydrodynamic radius. And beyond that, we can do two more advanced types of analyses. One is called the cumulance analysis, and that is good for what's known as a monomodal size distribution that is not too large in range. And so we can calculate an average RH and also a measure of polydispersity, and we'll see what that means a little bit later on. Another type of analysis is called regularization, and what that does is allow you to obtain a coarse size distribution all the way from sub-nanometer up to several microns without any types of fractionation. So that's really useful. You can use very little sample, takes no time, you don't have to worry about the fractionation, and you can get a coarse size distribution and measure a very large range of sizes. Third type of light scattering is called phase analysis light scattering, or electrophoretic light scattering, and I'll call it PALS. And what it does is measure the phase of the scattered light as you apply an electric field to the particle. It's moving. The phase changes in the scattered light give you an indication of the drift velocity. And so the ratio of the drift velocity to the applied field gives you a measure called the electrophoretic mobility. And once you also input the RH, the hydrodynamic radius, measured from DLS, you can calculate either the net charge or the zeta potential. The particular flavor of PALS that I'll be describing is called massively parallel PALS, or MP-PALS. And this is a unique technology that affords increased sensitivity and measurement speed, allows you to reduce the applied voltage so that if you have sensitive samples, they won't be degraded, and also allows you to make measurements in high salt buffer, to use fluorescing samples, and even measure in flowing streams. And we'll hear more about that later on. So these are the three essential flavors of light scattering, and let's examine what we can do with them. The first light scattering tool will be batch DLS, that is dynamic light scattering without any upstream fractionation. The two instruments that I'll be describing used to measure DLS are, first of all, the Dynapro Nanostar, which is a traditional cuvette-based dynamic light scattering instrument. It's unique in that it allows you to measure sample volumes as low as about a microliter. Very large temperature range. It has a dedicated static light scattering detector, which is useful for measuring molecular weights, and also allows you to incorporate an optical filter in case your particles fluoresce under the illumination by the laser beam in the cell. The second instrument is rather unique. It is a high-throughput dynamic light scattering plate reader, where we're making the measurements of dynamic light scattering in situ in standard micro well plates, either 96 through 84 or 1536 well plates, industry standard, will allow you to automate a very large series of measurements, including temperature scans. And another interesting feature of the Dynapro plate reader is that it has a camera on board, which will allow you to take photographs of the sample in the well in order to assess if there are bubbles there, or precipitates, or large contaminants, big particles floating around. The properties that DLS allows you to measure include, as I mentioned, size distributions, coarse size distributions, but over a very large range, in order primarily to assess the aggregation of the sample. You can also look at unfolding or the aggregation onset temperature. You can, particularly with the help of the automation, calculate the critical micelle concentration, critical micelle temperature, and also look at flow kinetics of self-assembly or dissociation, kinetics that are typically on the order of minutes to hours. So just to review what dynamic light scattering can provide, we measure the fluctuations due to Brownian motion. That gives us a diffusion coefficient. The cumulance analysis is suitable for monomodal samples to get average radius and polydispersity. Regularization analysis for looking at multimodal distributions, and I'll explain shortly just what those mean. And more importantly, through the Dynapro plate reader, DLS is a technique which is amenable to high throughput screening, so you can measure in an automated fashion many different samples hands-off. So what do those terms monomodal and multimodal mean? So the cumulative analysis allows you to look at monomodal distributions, and we see here one peak in size, so the x-axis is RH. We have one peak, and that's because our sample is monodispersed. So we can see a monodispersed sample up here, single peak down here, pretty narrow. If we have some aggregation or a polydispersed sample where the size range is not too large, we'll see a broadening of the peak over here. Okay, but it's still called monomodal. And so cumulative analysis will give you the average value as well as the width of the peak. Now, if you have a multimodal size distribution like the one shown here, we have both very small and large particles. That's called multimodal. It gives us two peaks in the size distribution. Okay. Now, as I mentioned, this is a coarse size distribution. So in order to be able to actually resolve these two different modes, we need to have about a 3 to 5 times difference in radius, which would be a very large difference in mass, in order to see those different peaks. Regularization also gives you the width of the peak. So if you have a both polydispersed and multimodal sample, like the one shown over here, then you'll have the broadening of the peaks as well. This is what we can learn from dynamic light scattering, and let's apply that now to some interesting nanoparticle challenges. So the first example would be a core shell microgel. You can see the core over here and the narrow shell. Okay. And this microgel was exposed to temperature scans. And so what we see is that if we have the core alone, which is these black dots, they undergo a phase transition right around this temperature of about 32 degrees, in which the core collapses. So it goes from a radius of about 110 to a radius of about 46 or 47. And so it is a thermoresponsive core. Once the shell is applied, we see now that the different types of shells that are applied do not undergo this large thermal response. So there is a little bit of collapsing due to the core collapsing, but overall the shell doesn't change nearly as dramatically. And so this is a very quick and rapid type of measurement that you can do in a cuvette-based system, just changing out your samples. Probably takes no more than 10-15 minutes to run each one of these samples. It's very convenient to do that. Maybe a little bit more than 10-15 minutes. So there's an interesting property of dynamic light scattering that is that when you're measuring the size due to the fluctuations, you're actually simultaneously measuring an effective type of static light scattering, the average intensity, and we can use the combined information from the size and the average intensity to understand if a transition is accompanied by some form of degradation, for instance, aggregation or dissociation, or is it simply a conformational change? And so in this example, which is actually a protein, you can see that as you go through the conformation change, the size is increasing, but the count rate, which is actually the static light scattering, is remaining the same. Now, if you remember the formula for static light scattering, it's proportional to molecular weight times concentration. And since we're not changing the concentration, if the intensity does not change, that means that the molecular weight is not changing, and therefore there is no degradation of the sample, simply a conformational change. In other cases, you'll see that the count rate increases, and that indicates that there is aggregation, or it decreases, and that would indicate either dissociation or precipitation of the sample. Now, it's really convenient to be able to make these measurements in an automated fashion, and that's why we have the Dynapro plate reader, where the measurements are made actually in the plate. There's no liquid handling associated with, say, sampling the material out into a separate instrument and then returning it to the plate. It's all made directly in the plate. And the software, the dynamic software, which is used in the analysis, allows you to create a heat map, as shown here, for this 96-well plate. And you can set up the colors to give you a quick visual indication of the aggregation state of the sample. And so in this particular case, we can see how those colors were coded. So the red indicates a nice monomodal sharp distribution, which would correspond to a primarily not monodisperse sample, perhaps with low aggregation. The blue was set up to indicate moderate aggregation. And then finally, the black was set up to indicate very large-scale aggregation. You can't read the numbers down here, but they're actually giving sizes of a micron. So the monomodal sample would be on the order of a few nanometers, whereas these aggregates are several microns, and so obviously very, very large-scale degradation. This whole scan takes less than 45 minutes, so it's very convenient to set up, walk away, and come back and find this heat map ready to go on your desktop. Another interesting application of dynamic light scattering for nanoparticles was published by Dennis Lung and colleagues in the APS journal. And here they're looking at nanoparticles which are produced by the acoustic milling of drug solid. So this is a small molecule drug, and using this resin acoustic mixing technique, which actually takes place in a 96-well plate. You put in your drug solids, you put in some milling particles, some particles that bounce around under the resonant excitation and break up the drug solids, and also a polymer, which is used to coat the particles as they form and prevent them from aggregating. And so the idea was to be able to quickly establish optimal processing conditions, what is the right amount of surfactant to put in, the right amount of time to do the milling, and so forth. And so they were able to set this up very easily. The waste and acoustic mixing takes place in 96 well plate. Small aliquots were periodically pipetted into the 384 well plate, and then the measurements here are shown in the heat map. And using this, we're able to determine excuse me, the particle size of the resultant drug nanoparticles as a function of the surfactant and surfactant particle ratio, and also dissolution time. So as opposed to doing a whole series of one-by-one measurements, this just made it extremely easy to go forward and establish the optimal processing conditions. So really, the Dynapro DLS plate reader is an optimal productivity enhancer, because you can set up all these different things, walk away, do the temperature ramps, run many, many different measurements, come back later on and have it all set up for you on the heat map. So another interesting example of using dynamic light scattering. So following the critical micelle temperature of a polymer, this was an elastin-like polypeptide diblock copolymer, and at a particular temperature, about 41 degrees, it assembles into weak micelles. Now, many people would actually make this measurement using fluorescence, but the advantage of using DLS to make the measurement is that you're looking at a quantitative physical property which has some meaning. So rather than fluorescence, which is loosely correlated to some change in the environment, you're actually seeing the size change from the size of the unimer here at about 8 nanometers. So formation of micelle up at about 30 nanometers. And then once you reach about 50 degrees, you have wholesale aggregation. Now this could actually be done in the plate reader as well, in the Dynapro plate reader, and you would get full-scale automation of measurements of the critical micelle concentration, critical micelle temperature. You would get the size from the dynamic light scattering, and you'd also get changes in conformation associated with count rate. If you need to go over a larger temperature range than you can accomplish with the plate reader, then you can use the NanoStar, which again uses single cuvettes, and you'd make these measurements one by one. So the dynamic light scattering tool for nanoparticle characterization allows you to determine size and size distributions. The thermal behavior allows you to do high throughput screens, in particular to establish optimal reaction and processing conditions, and you can quickly assess the solution properties with just a few microliters of sample. Next tool I'll describe is MP-PALS. And so MP-PALS utilizes the instrument known as the Möbius, which measures both dynamic light scattering and electrophoretic light scattering. It does include a flow cell so that you can automate it using an autosampler. You can connect it to an online separation technique, or you can load it manually. It offers an accessory known as the Atlas, which pressurizes the cell and allows you to make zeta potential measurements in high salt buffers. Also has an optical filter in case your particles fluoresce. If you have volatile solvents, it's a closed system, and you can operate in both batch and flow modes. The properties that this tool measures are electrophoretic mobility, size and size distribution through DLS, and the combination of the mobility and the DLS gives you the charge or the zeta potential on your molecules or particles. Applications include colloidal stability, so the zeta potential is most often used to promote the stability of the colloids, but this charge measurement also allows you to look at things like the DNA polymer NP ratio, which can be optimized for transmembrane delivery. Brief schematic, we have electrodes applying a voltage, the sample drifts between them, and the light scattering gives you this, hits this array. This is an array of 31 independent simultaneous measurements, unlike traditional PALS detectors. And that gives you higher sensitivity, reduced measurement time, so you can actually measure it, make relatively short measurements. And this technique is impervious to fluorescence or saturation because it's not using an APD or other detectors which are typically used for photon counting. And the one example that I show is this PAMAM dendrimer, which is measured over several dendrimer generations, from 3 all the way up to 7, and also in a few different solutions, which included some really high ionic strengths. Again, using the atlas, it allows you to apply pressure, and that eliminates the bubbles that are caused by electrolysis, allows you to make very accurate measurements at very high ionic strengths. And you can see that this was all set up automatically through an auto sampler, and so you just set up your samples and tell it to go, come back, and you get all your measurements later on. So MP-PALS instruments do, first of all, everything DLS does because they include a DLS detector. Allows you to screen formulations for optimal zeta potential in order to promote stability, look at chemical modifications of the surface charge, and finally, because the measurements in the Möbius and the MP-PALS detector are made simultaneously with the DLS measurements, you can monitor any type of degradation that occurs as you apply voltage to your sample. Next tool will be FFF-MALS. And so FFF-MALS takes our multi-angle light scattering detector and some concentration detectors, for instance, refractive index and UV, and connects them to a standard FEC system, size exclusion chromatography, or GPC. System will include a DON or mini-DON, which are the MALS detectors provided by Wyatt. Refractive index detector is the Optolab T-Rex. You can embed inside the MALS detectors a DLS module, and so that you're actually making the dynamic light scattering measurements and the static light scattering measurements in the same flow cell. This is advantageous relative to adding on another detector downstream, which will not be measuring simultaneously. And finally, if necessary, we'll be using the UV detector for the liquid chromatography system. Properties measured by FFF-MALS include absolute molecular weight and size, a very large range of molecular weights from 200 Daltons all the way up to gigadalton, a large range of sizes, both the RG measured by MALS, as well as the RH measured by dynamic light scattering. And these allow you to characterize small aggregates and oligomers that will be separated by FEC. We can analyze conjugated materials, for instance, copolymers or proteins conjugated to carbohydrates. By combining the information of the two types of sizes, the RH and RG, we can learn about the conformation of our particles. And finally, the asterisks here relating to the range are here to indicate that the range is primarily limited by what you can separate on FEC. Some of the larger sizes here, for instance, 240 nanometers in size that you can measure with DLS, you would have to go to a different separation technique called FFF that I'll describe later on. And so here's an example of why it's really important to use multi-angle light scattering if you're characterizing your macromolecules using FEC. FEC, contrary to popular belief, does not separate based on size, based on molecular weight. It separates based on hydrodynamic radius or hydrodynamic volume. And so if we have a standard, for instance, a polystyrene standard shown here, they'll elute at particular times which correspond to their radii, shown here. Okay. And then if we have the unknown sample, our particular sample, it'll have some type of distribution. And if we measure the through dynamic light scattering the hydrodynamic radii, we'll see that, again, that the radius measured for the standard and the radius measured for the unknown sample are identical at identical elution times. And that's because the separation is based on size, so it makes sense. However, the polystyrene standard might not have the same conformation as our sample. And therefore, even though at a particular elution time they would have the standard and the unknown sample would have the same hydrodynamic volumes, they could have different molecular weights. And that's shown here. The multi-angle light scattering measures molecular weight, and they're actually different for the standard and for our sample. And so just based on retention time, we don't have a good indication of molecular weight. And because accurate mass does matter, you wouldn't want to try to knock down a building with a beach ball. You need to know the correct molecular weight, and you need the right tool to measure it, that being multi-angle light scattering. Here's another example of why multi-angle light scattering is important in FEC. Again, if you're using retention time and column calibration and just inject different amounts of sample, a 10 microliter injection and a 50 microliter injection, you could have different elution times, which would, based on column calibration, indicate that you have different molecular weights. Of course, that's not true. You're just injecting different amounts of the same material. The light scattering shown here shows that, in fact, you do have the same molecular weights, whereas the column calibration would show that you have different molecular weights. And that's reflected here in the table. So these are the peak molecular weights for the two determined by column calibration, quite different, versus by multi-angle light scattering, the same. Here's an example from the protein world. So again, we have our standards, the BSA monomer and the BSA dimer, and they elute at the times that you would expect for nicely behaved globular proteins. But hemoglobin, which has actually about the same molecular weight as the BSA monomer, elutes much later. And based on calibration, column calibration, you would expect it to have much lower molecular weight. This is, of course, incorrect. The light scattering gives you the right answer. It shows that it has a molecular weight of between 52 to 62 kilodaltons. And actually, the shoulder that you see over here with decreasing molecular weight corresponds to a slow dissociation of the hemoglobin, which is nominally a tetramer, dissociating into dimers, and that reduction in molecular weight shows up in the light scattering. So actually you get a lot more information rather than just the molecular weight. You have information here on dissociation as well. Traditional SEC assumes that your calibration references and your samples are ideal. They have the same conformation, same density, there's no different column interaction, and that your chromatography is reproducible, but those are quite often incorrect, and therefore your column calibration will be incorrect. The multi-angle light scattering does give you the right answers in those cases. Here's an example of looking at a very broad polymer. So first of all, here we also injected a few different volumes, and there was no shift or change of molecular weight with concentration. We have the RI signal here, we have the light scattering signal over here, and you can see that, basically the distributions are the same. Here are the molecular weight distributions over here. Try this one. At the three different concentrations. And you can see that they range from over 1 million and down to about 1,000. And then finally we have the monomers. We have the monomers of this polysaccharide. And they have a molecular weight of just 214 grams per mole. And again, we can detect that. Actually, these very small polysaccharides don't scatter much light, so you need a bit more concentration to make the measurement. That's a 200 microliter injection. We couldn't detect it with 50 microliters, but very large range of measurements that you can determine accurately using multi-angle light scattering. And so yes, there is a very large range. All of these sizes are important, both large and small, and we like all of them. Another important application of SEC-MALS is analysis of conjugates. And so those conjugates might be pegylated proteins, protein DNA complexes, polymer peptide assemblies, or copolymers. And the way that we do the analysis is through a 3-detector system, which uses a UV detector and a refractive index detector to establish the ratio of the two different species in the complex. And finally, the multi-angle light scattering detector combines with that additional information to determine the molecular weight of each of the species or each of the components in the complex. And so this is perhaps most easily explained using an example of a carbohydrate conjugated to a protein. The UV gives you the protein concentration. Refractive index gives you the total concentration of protein plus the modifier. And then the combination of UV and RI will allow you to determine independently the concentration of the protein in the complex and of the modifier in the complex. Finally, adding in the light scattering data, you get the molecular weight of the protein part in the complex, the molecular weight of the modifier, and finally the total molecular weight, again, which are shown here across this peak. And so this is can be used quite effectively in looking at the efficacy of reaction. So here's a pegylation reaction, and you can see that the peaks corresponding to unreacted PEG, unreacted protein, the heterogeneous pegylated protein, and finally the aggregates, can all be identified, and they can be identified through that 3-detector technique, which tells you for each of these species how much protein you have and how much of the PEG you have. So to summarize tool number 3, FFF-MALS, including FCC with online DLS, we get absolute molar mass and size in solution, overcoming the limitations of the column calibration standard GPC or SEC, which might be due to non-ideal samples, which have different conformation or column interactions than the standards. You can get positive identification of reactive and unreactive species. Something which I haven't shown, but you can also address is decorated nanoparticles. That again would be through the conjugate analysis, and then all these other types of conjugates that you can analyze, polypeptides, DNA, protein, et cetera, et cetera. If you're interested in looking up some peer-reviewed articles that explain the use of FFF-MALS for these applications, you can go to our bibliography, online bibliography, where you can type in search terms and find hundreds, thousands of articles citing our instruments which have done these measurements. Next tool I'll describe is FFF-MALS. So FFF field fill fractionation is a separation system, similar in concept to SEC, but it does work differently, and it is superior in many ways to SEC. It's a little more complicated. So the system would include a standard HPLC stack, our standard multi-angle light scattering detectors, and finally the clips, which is an additional component that includes a separation channel over here, which would replace the column in an SEC separation. It does integrate with industry standard analytical HPLCs. And in fact, the dual-tech model of field fill fractionation will allow you to automatically switch between FFF and SEC mode, so you can run your samples either on the column or on the FFF separation channel. The properties that you measure with FFF-MALS are similar to those that you measure with SEC-MALS, except you can cover a much larger size range. So you can measure a range of sizes from 1 to 1,000 nanometers. Very briefly, this is the FFF channel, separation channel. It's an open channel. You see that there's no column packing, and there is 2 different flows. There's a cross flow, which goes that way and pushes the particles against a semi-permeable membrane. There's a channel flow that goes that way. Based on diffusion, the particles will end up at different heights above the membrane, and because there is this parabolic fill profile, the higher things are up in the channel, the faster they come out, and therefore the smaller particles, which diffuse higher up, will come out first. The larger particles will stick around longer. This is opposite to what you expect for SEC, so it looks a little bit different. One of the great advantages of this is that you can actually do a very nice separation of the larger sizes. So because there's no stationary phase, you have very little shear, you have very little adverse interactions with all that surface area, and you can get resolution which is good as or better than size exclusion, as well as optimize for different separation ranges simply by adjusting flow rates or some geometric parameters of spacing size. Here's an example of separating on FFF. This is some standard latex particles. Very often latex particles are measured by dynamic light scattering, but there's no way that you'd be able to get this type of resolution with dynamic light scattering, with batch dynamic light scattering. Here you can see beautiful baseline separation of particles, which are not too different in size. And because we're measuring the size with light scattering, we can actually see here the amount of polydispersity. So these particle standards are not extremely monodisperse. They have some polydispersity, and we can pick those up using the separation technique. Here's an example of separating a VLP, 2 different VLPs, one with 180 subunits and the other one composed of 240 subunits. Total sample injected here was just 2.3 micrograms. And again, you can see beautiful separation to determine the different populations. Using online MALS, we can get both the molecular weight and the size. And of course, this type of separation and characterization would not be feasible with any type of batch measurement, such as dynamic light scattering or nanoparticle tracking analysis. They just don't give you the resolution. So if you really need the excellent resolution characterization capabilities, FFF is the way to go. This is, again, looking at a process. So we have some completely reacted VLPs and some incompletely reacted VLPs separated. And here we've looked at a VLP which was packed with some DNA using that conjugate analysis algorithm with 3 detectors to determine the molecular weight of the DNA, of the protein, and finally of the entire VLP. So you can characterize very well the encapsulated DNA mass. Here's an example where we're looking at a nanolipid complex which was soaking up some microsilver particles in the solution and examining as a function of the process conditions what is the concentration of the nanolipid and the concentration of microsilver in solution on the ultimate size and size distribution of the complexes. And so again, doing an FFF separation and measuring the size using online light scattering, we can get beautiful distributions, very accurate distributions, and you can I don't show repeatability data, but these are actually highly reproducible. And so you can discriminate between sizes on the order of 1 or 2% difference, something which would be literally impossible to do with dynamic light scattering. But FFF gives you very accurate and high resolution separations. The addition of the online light scattering gives you the accurate characterizations. Now, as I mentioned, you can use the combination of dynamic light scattering, looking at RH, and multi-angle light scattering, giving you RG, to learn something about shape or conformation. So the ratio of those two is known as the shape factor, or rho. And what we see here is the ratio, how the length of the major axis in the ellipsoid, B, varying between 2 and 100 nanometers, versus the minor axis, A, 1.5 nanometers, gives you different shape factors, rho. So this is a kind of a calibration curve, which is calculated theoretically. And if you look at 2 different particles separated over FFF, and rho being measured with online MALS and DLS, we can determine that the blue particle has a shape factor indicating a solid homogeneous sphere, whereas the red particle is more rod-like and elongated. So again, we can get shape information from this combination of online MALS and DLS with FFF separation. Now, liposomes are really popular for using in drug delivery. I think probably half the people I spoke to at CRS were interested in using liposomes for drug delivery. And if you are encapsulating a drug in your liposome and measure the sizes using DLS, you'll get no difference in radius. The 2 different liposomes filled and empty will have the same size. So there's no information to be had there. On the other hand, if we utilize this technique of separation with FFF and online measurement of both MALS and DLS, we can get some useful information. So again, RH measured by DLS on these separations are identical for filled and empty, just like in the batch measurement, but RG is different. So RG is actually related to the average, the mass average position of the mass in the particle. And because one is filled and one is empty, the average position of the mass will differ between the two, and we actually see different values for the empty and filled liposome here. And we can understand that theoretically. The empty sphere would have a ratio, a shape factor of about 1. The solid sphere would have a shape factor of 0.77, and these show up very nicely in the graphs. These are, in fact, the ratios that we get. And so, yeah, it is important to know what's inside your particle when you're delivering something important as a drug. And so using the right type of characterization technique to reveal that structural information can be really valuable. Another thing that we can do with FFF-MALS is particle counting. And so that uses the analysis, which I won't describe right now, but it does use basic light scattering theory, first principle light scattering theory, to determine the actual numbers of particles in solution, the number density. And in this comparison, using an imaging technique versus the light scattering technique, we can see very similar values in terms of what we get from the counting, from the number density analysis provided by the Astra software. So again, a very quick and effective way to measure size distributions, not just in terms of concentrations, but also in terms of numbers of particles. And in this example, we took that same core shell microgel, and this would be the core, and took those number distributions, differential size distributions. So the core is about 53 nanometers in size. The non-degraded shell is about 55 nanometers in size, and the degraded shell here is about 58 nanometers in size. And so you can see these very small shifts, but they're very clear and very accurate. This is the same thing for a different type of shell. And so the shell degradation leads to an increase in the particle size, but not in RH. So if we were to measure RH, which is the envelope of these particles, it doesn't change at all. That tells us that there's an internal shift in the mass, the core is swelling, and we can see very small differences using this technique. Finally, we can take our Möbius, the PALS, and put it online with an FFF separation and measure the electrophoretic mobility of different sizes of particles. And so what we've done here is we put it online with separation of some polystyrene latex spheres and measured the mobility of the three different sizes. So this is an interesting way to characterize a mixture of particles where you're actually separating them first by size, then determining the zeta potential or the mobility of each one. So FFF-MALS really is actually a really important tool for characterizing drug delivery nanoparticles. It's a primary tool for full characterization of the size and conformation, primary tool for characterizing any type of complex fluids, which might include macromolecules plus submicron particles, free and bound or encapsulated drug. It's a primary tool for establishing molar mass and size of macromolecules that don't elute well on SEC due to poor column interactions. Will validate and analyze your vesicles, and finally you can, downstream of the FFF separation, you can add the PALS detector to get high-resolution distributions of zeta potential. The last tool I'll describe is CG-MALS, which stands for Composition Gradient Multi-Angle Light Scattering. This is a technique which actually has lots of applications in the arena of biomolecular interactions. It is a type of stop flow system, and the applications that I'll describe here that pertain to drug delivery nanoparticles will pertain to the reaction kinetics. And this is one way of using it because it's a stop flow system. It's not a super fast millisecond stop flow system, but it can allow you to characterize kinetics from seconds and on. So very briefly, the Calypso system, which is the composition gradient component, consists of 3 computer-controlled syringe pumps. Those solutions are run through filters and injected into the light scattering and concentration detector. For the example I'll show you here, we won't have a concentration detector, just the light scattering detector will suffice. And this was, again, a microgel. Okay? And so the microgels were exposed to periodate by combining them in the stop flow technique. And if you look carefully at the molar mass that's calculated using multi-angle light scattering and the size using the angular dependence, what we see here is a very rapid decrease in molecular weight over about 2 orders of magnitude. So we're measuring here about 2 orders of magnitude decrease in molecular weight due to the fission of the cross-links by the periodate. But if you look at the size, which is the red over here, you see that initially actually increases a little bit, and then very, very gradually decreases. So from about 165 nanometer radius to about 150. A very small change, even though we have 2 orders of magnitude decrease in molecular weight. What does that tell us? That tells us that, as is illustrated here in these representations of the particle, even though you're thinning the structure by cutting those crosslinks and material is coming out of the nanoparticles, hence the large decrease in molecular weight, the actual overall growth structure is not changing much. You're keeping the same size. So you can get interesting confirmation of the structural changes due to the degradation through this analysis by time-dependent multi-angle light scattering. Multi-angle light scatter is also used to measure the uptake of a protein into a nanogel. And so here the nanogel was exposed to different concentrations of cytochrome C, which was the model protein, ranging from 10 nanomolars here up to about 100,000 nanomolars here. And you can see again, looking at molecular weight, which is the blue, a very large increase in molecular weight by about 100-fold. So a very, very small molecular weight when there's very little protein, up to a very large uptake in molecular weight. But the size, again, is decreasing here. So in this case, the size is decreasing from about 225 nanometers over here to about 200 nanometers over there. Why is that happening? If the molecular weight is increasing so rapidly and in such large numbers, how is it possible that the size is decreasing? Well, it turns out that this is a very sparse microgel, and the strands are held apart by the charge on those strands. That charge is also what's drawing in the protein, which has an opposite charge. And so when the protein comes in, it neutralizes that charge and allows the microgel to collapse, and that's the change in size that we see here. So another interesting type of structural characterization in addition to learning about the uptake properties and encapsulation that we can obtain with light scattering. So CG-MALS, besides the biomolecular interactions, will also allow you to look at the kinetics of reactions, drug loading, through changes in molecular weight and size, on timescales of seconds to hours, and you can readily automate a series of preparative conditions, for instance, if you wanted to look at CMC. And so here are the answers to our questions. How are these different light scattering techniques, depicted by the different acronyms here, used to address the different questions that come up? And so if you're interested in pursuing more, I encourage you to visit our bibliography, where we have over 11,000 peer-reviewed publications citing the use of Wyatt instruments in exactly these types of applications and many more. And finally, if you're interested in learning more, I encourage you to visit our website. You also have links. You have some links shown in a panel on your browser, I believe. And with that, I would like to thank you for your attention and hand it back to Judy.
Thank you, Dr. Sohm, for that informative presentation. It is time for Q&A, so if you have a question you'd like to ask our speaker, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the Send button. We'll answer as many of your questions as we have time for, so let's get started. Our first question is, what is the best way to discriminate between empty nanodiscs and nanodiscs loaded with protein?
Yeah, so exactly the conjugate analysis that I described earlier on in the context of SEC-MALS. Nanodiscs typically can be used with SEC-MALS to get a nice separation. And a nanodisc is typically composed of a protein ring encapsulating a lipid bilayer. So here's our protein and modifier. And using the 3-detector technique, you can get the molar mass of the protein and the molar mass of the modifier. If you have an additional protein encapsulated in the nanodisc, then obviously you'll see a change in the protein molecular weight relative to the modifier molecular weight, and that should give you your answer.
Next one is, how can I monitor particle aggregation over time?
So it depends on what causes the aggregation. Certainly a very simple way to do the measurement is to put your sample into the dynamic light scattering cuvette. If the aggregation is rapid, you would just leave it there and look at the change over time. If it's a slower aggregation, then you would just extract aliquots and periodically put them into dynamic light scattering. If it's an aggregation that is induced, for instance, by the addition of some reagent, then you could use the CG-MALS system to do that combination, inject the mixture into the light scattering detector, and stop the flow, and again monitor the change in size and molecular weight through light scattering.
Why is there a large difference between the expected molecular weight of a protein and the value measured by light scattering?
So there are a few possibilities for that. One is that the protein is in a different oligomeric state than you expected. So if the expected molecular weight corresponds to that of a monomer, it's possible that it is actually oligomeric in solution, and light scattering will measure that oligomeric state in solution. Another possibility is that it's glycosylated, or some other type of post-translational modification occurs. And again, if your expected molecular weight corresponds to the neat protein without glycosylation or PTMs, the light scattering will measure the entire molecule with the glycosylation or the additional modification, and will give you, again, the actual solution state of that protein.
How can I measure the amount of unbound drug in my sample of drug-carrying nanoparticles?
So I'd say a good way to do that is to do a separation. Depending on the size of the nanoparticle and its properties, you might be able to do the separation on SEC. Otherwise, you could do the separation on FFF. And any unbound drug will clearly be separated and will elute at a very different time. So on SEC, you should be able to use the light scattering concentration detectors to determine which is which, what is the state of the unbound drug, and what is its species. Sometimes if you have a large particle, a large nanoparticle, encapsulating a small molecule drug, you would have to do that separation on field flow fractionation. But if you remember, the field fractionation configuration has a semi-permeable membrane, and the drug could actually escape through the membrane during the separation. But you can monitor the eluent of the crossflow. So you can put a concentration detector on the crossflow to see if any of that drug is escaping, and I actually expect to be able to publish an application note describing that sometime soon. So those are good ways. You do the separation and characterize each one individually. You can use downstream of FFF and SEC not only the standard light scattering detectors, you can use concentration detectors, you can add in an online fluorescence detector. So there are many types of characterization tools that you can apply once you've done that initial separation.
My nanoDDS system is biodegradable. How can I characterize the degradation rate?
Yeah, so a good way to do that is to utilize the CG-MALS system. You can have in one of your solutions would be your sample, and the other would be a reagent which causes the degradation of effect, something that creates an environment similar to the physiological environment, or some other bioenvironment which leads to degradation. Once the stop flow is affected, you've combined the two solutions, injected them into the light scattering detectors, and the flow stops. Then the light scattering will measure the change in molecular weight, change in size. So if you have aggregation, or more particularly in the example that we looked at, you have some type of dissociation due to that additional reagent, that will impact the size. You can measure size through multi-angle light scattering. Simultaneously, you can measure through dynamic light scattering. And that combination will give you lots of information on how exactly and at what rate the degradation occurs.
How do different buffers, including cell culture medium, affect the radius and beta potential assay results?
That, of course, depends a lot on what the particular cell culture medium is and what the sample is. If you're looking at dynamic light scattering where there's no separation, then anything present in any significant quantity in the medium needs to be well differentiated from the sample of interest. So if you remember the regularization analysis, you need a separation of approximately factor 5 in radius in order to get good separation. On the other hand, if you can do a separation, and that's not always an option with cell culture medium, but if you can do a separation, then you can, and the components of the medium are quite different from your sample. For instance, your sample is a large particle and the medium is just smaller molecules, then you can separate them out very nicely and characterize each one individually. Now, if you need to look at the kinetics of something going on, then you might extract the aliquots periodically. That's a good option. Or just leave them, for instance, in the dynamic light scattering detector over time and see how the changes occur.
are almost out of time, so this will have to be our last question. What is the limitation in size measurement of your DLS equipment?
So we typically state the sizes in terms of radius, and it also depends on the particular instrument. The plate reader goes from about half a nanometer up to 1 micron. The cuvette-based instrument, the Nanostar, goes from 0.2 nanometers up to 2,500 nanometers, or 2.5 microns.
I would like to once again thank Dr. Sohm for his presentation. Do you have any final comments for us?
I just want to mention that we will be collecting all the questions and answering them in a document, and we'll be posting that document on our website, also sending them out to all the attendees. So thank you for your attention. I appreciate your presence.
Thank you again. I would also like to thank our sponsor, Wyatt Technology, for making today's educational webcast possible. Just a reminder, if you wish to obtain the credits for today's session, please click on the Get Your Free CME CE Credits button located in the lower left of your screen. This will take you through the process to obtain your certification. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.